why don't we try and get started? And again, thanks so much everybody for joining. I think these monthly teleconferences have generally been very well attended, have been at a very high level. And I think it's very much fulfilling the mission and the goal that we had within INTBEER to uh, promote international collaborations, et cetera. And I know that we have many projects already underway that have grown out of these discussions, right? So today, and Jussi, you changed the topic. If you can go ahead and change your screen, the title that I have that was initially distributed last week turned out it's not going to be the actual title that you're going to talk about, but nonetheless, I'm a big fan of all the work that you've done over the years, and always very interested in knowing what you're looking at. So you have this new study called OVERCOME-TBI. Would love to learn
Yes,
more about that.
yeah, thank you so much. So I will start sharing. I hope you are able to see my
We
slides.
can see your screen. Yeah.
Yeah, good. So thank you again for the invitation, and I will, in the next 20 slides, I will introduce our new study that we will be starting in
Amy, are you
Turku,
recording
Finland.
or should I record?
I'm recording, thanks.
Okay, great.
Yeah.
Good.
Great. So, and like Ramon said, the study is called OVERCOME-TBI, and it's an abbreviation from long-term recovery and microbiota gut-brain axis disruption after TBI. So, a couple of words about the background. So, a more detailed characterization of the disease phenotype is crucial for improving TBI management and predicting outcome in individual patients. And although TBIs in the elderly have increased rapidly, most victims are still children and working age adults. Of course, the incidence in older adults is growing in fast pace in the Western world, but still, like said, this is mostly a disease of the working age adults. And despite of the significant impacts on health, many fundamental questions about pathophysiology, diagnostics, and treatment remain poorly understood. The main reason for this is the enormous complexity of individuality of TBI despite its initial severity, where no two injuries are identical. So the key to develop effective therapies for TBI lies in better understanding and identification of precise mechanisms underlying pathology associated with TBI. Recently, a commission in the Lancet Neurology on TBI highlighted the need for novel approaches to identify and fight the devastating long-term consequences of TBI. So there's a call for more research on the relationship between TBIs of different severities and their long-term effects and likely association with neurodegenerative disease. In a significant minority, TBI has been identified as a trigger of progressive deterioration and risk for chronic neuroinflammation and or subsequent neurodegenerative disease. There's increasing evidence that immune system plays a significant role in TBI pathogenesis. TBIs of different severities can lead to chronic neuroinflammation, which can be potentially detected on head positron emission tomography and or by blood biomarkers, and which can persist years after TBI. The basic assumption is that inflammation is associated with almost all brain injuries. Given the far-reaching influence of the gut microbiome on the central nervous system, there is also growing interest in studying the role of microbiota gut-brain axis in patients with TBI. So the microbiota gut-brain axis can be understood as an integrated physiological model encompassing afferent and efferent signals. And as a result, there's a growing interest in the local and systemic impact this communication pathway has on host health and disease burden. So this is the hypothesis behind the study. TBI results in neuroinflammation and microglial activation. Hyperactive immune response results in increased gut permeability and altered microbiota. And systemic response is a result of gut impairment, and peripheral pro-inflammatory cytokines exacerbate or maintain neuroinflammation. So Virginia Newcomb showed in her paper a couple of years ago that GFAP and NFL level can remain elevated months to years after TBI and exhibit different temporal profiles, which correlate with microstructural injury of the brain, and it's also associated with accelerated brain aging and contemporaneous quantitative diffuse integral MRI and functional decline. So this is a hypothetical kinetics of GFAP and NFL and cytokines and chemokines in patients. So I was about to present the hypothesis of the study. So blood-based protein metabolomic and inflammatory biomarkers can identify patients with TBI who exhibit signs of progressive neurodegeneration. This means progressive brain aging, functional and/or cognitive decline in the chronic phase of TBIs. And patients with progressive brain aging, functional and cognitive decline exhibit signs of both neuro and intestinal inflammation, as well as altered gut microbiota and intestinal permeability in the chronic phase of TBI compared with recovered patients with TBI and control patients. And in this case, it means patients who have sustained orthopedic injuries without TBI. So this will be a cross-sectional study that also includes some longitudinal assessments. So we have two old legacy TBI cohorts that we have studied here in Turku. And they include patients who have a history of TBI sustained 1 to 4 years and 11 to 40 years earlier. And in the TBI care study, we have biomarker and imaging data on 160 patients, and from Becker's TBI study, we have dense blood samples or densely sampled blood samples and imaging from 180 subjects. And we know that in these cohorts, there are patients who have had worsening symptoms over the years and who have stable symptoms, and patients who have recovered. And we will now invite the patients for new assessments, and the new assessments form the basis of this study. So, the both studies include serial biomarker sampling and subacute and follow-up visit head MRIs, and they both include also around 40 orthopedic controls. So the primary outcome measures will be cross-sectional and longitudinal biochemical neuroinflammation burden, measured by blood-based proteomics, metabolomics, and lipidomics in patients with TBI compared with trauma controls. In other words, orthopedic control patients. And longitudinal functional outcome measured with GOSI in patients with TBI over time. And cross-sectional changes in microbiota. We will analyze DNA from stool sample microbiome and intestinal wall microbiome assessed by deep shotgun metagenomics approach in patients with TBI compared with trauma controls. And brain inflammation burden on head positron emission tomography using PK11195 radioligand in patients with TBI compared with orthopedic trauma controls, and also positive comparison group with MS patients. And secondary outcome measures will be changes in brain white and gray matter microstructure. We have serial MRIs from these patients over the years, and now we will obtain new ones. And we will also assess whether patients who are symptomatic have accelerated brain aging. So this is the study flow chart. First, here are TBI patients and orthopedic controls. We have done this part already. The primary screening, we have identified those who are alive, thanks to Finnish registers. And we will contact the surviving TB care and the packaged TBI patients, and they are invited for a follow-up visit. And of course, those who are unfit, unwilling, or unable to participate will be excluded. We will have neuro and gastro assessments, blood and stool samples, head MRI, and PET on those who are willing to go for PET scanning. And from these assessments, we will obtain data for neuro studies. Then we will screen patients for gastroenterological assessments. Qualified gastroenterologist will do it. And then we will perform a standard colonoscopy, pinch biopsies for histology, microbiota, and metabolomics and lipidomics, and also assess fecal calprotectin. Then we will have the multiple sclerosis patients, and they will be recruited from the outpatient department visits. We will obtain blood samples, head MRI, and PET. And the inclusion criteria for TBI, all those who are alive and give their consent will be included. And secondary inclusion criteria for TBI and ortho controls and regarding gastroenterology, all those who are alive and give their consent will be included. And we will include those patients with MS who fulfill the McDonald 2017 criteria, and they exhibit significant white matter lesion load. Exclusion criteria, TBI sustained outside the original studies, and those patients who have contraindication for head MRI will not, of course, undergo head MRI. Instead, they will undergo the other assessments. And for controls, those who have suffered TBI or any other brain disorder after the original studies will not be included. Then there are lots of exclusion criteria for gastroenterological studies. I will not go through them to save time. And there are also exclusion criteria for PET imaging. So we will do many assessments. The patients will undergo neurological and cognitive testing, blood sampling for blood-based biomarker, head MRI, colonoscopy. And up to 60 patients with TBI will also undergo PET imaging using PK11195 radioligand. And patients with orthopedic injuries will also will serve as controls and will undergo similar assessments. And we will have those MS controls. And all patients will receive compensation for participation. This is just to make sure that we will be able to recruit enough patients since we have quite invasive assessments and PET imaging. Fortunately, Finnish patients are usually quite willing to participate in medical studies, but we'll see. So we expect that 90% of patients to be included in the gastroenterological investigations. They will get free colonoscopy, and since they are working age or nowadays older adults, I would assume that it's a benefit that maybe many want to have. And I assume that a stool sample will be obtained from all participants. So the aim is to investigate the inflammatory state and permeability changes in the intestine to assess the overall inflammatory burden together with fecal microbiome profile, blood biomarkers, and PET, and to correlate the findings with neurological disease progression. So this is an observational, cross-sectional, and on the other hand, partially longitudinal study involving a real-life approach. So the data will not be collected in a way that groups would be balanced in terms of demographic variables. The maximum number of subjects recruited is 164 TBI, 34 orthopedic injuries, and 10 for MS. And regarding the PET sample size, in the healthy brain, TSPO PET typically shows low and uniform uptake in most brain regions. And TSPO, the translocator protein that is expressed in microglia and astrocytes, it's primarily upregulated in response to neuroinflammation. And based on the previous experience with PET ligands, it is estimated that 10 subjects per group are sufficient to detect 50% difference in the PK1119. 5 uptake between the study groups with a 9% power at p value of less than 0.05. So this is the summary. Sorry about the busy slide. So we have a permission from our ethics committee, and the study has been registered at clinical trials. This is our research group here in Turku. Olli Tenovuo founded the group already in late 90s, and fortunately he's still with us and serves as a senior researcher and a mentor for us younger guys. So thanks to these people for the upcoming study, and I will stop here. Thank you.
Well, that's a very exciting project. It's terrific that you're launching on that. I think the work that Ollie started and you've been part of for many years now there in Turku has been hugely influential, and this seems like it's going to continue along. There was a question from Cheryl on the chat. Cheryl?
Sure. Thanks, Ramon. Jussi, this is really fantastic. I'm wondering if you have collecting data on how people's diets may have changed after their injury, and if that might explain some of if there's microbiome changes.
Yes, thanks, Cheryl. This is really important question, and on the study visit, we have a gastroenterologist who will conduct a questionnaire regarding their diets and also the use of antibiotic treatment over the years. So those will be accounted for.
Wonderful. Thank you.
There was also a question regarding the proteomic and metabolomic approaches that you were planning to use. If you'd elaborate a little more on that.
Yeah, thank you. So yeah, again, important question. So we have published a couple of papers with Matei Oresic's metabolomics group, and we have some candidates, of course, that we will definitely assess, but we will mostly use untargeted approaches for proteomics and metabolomics. Any other questions?
Can I follow up
David?
on that?
Yeah, there was a question to Dr. Winston.
Yeah, sorry. So the problem with the untargeted approach, it's not that quantitative. And so that could be a real issue in the long run. Just thoughts, that's all. And clearly a more targeted approach would give you quantitative data. So that's just something to think about.
Yeah, thank you for this remark, and I will discuss this with our metabolomics and lipidomics experts.
But in principle, you will have biological fluids that even though you have things that you're putting your nickel on for this project, there will be the possibility with additional funding, et cetera, to do further analyses. You'll have biological fluids left over.
Indeed.
David, you had your hand up.
Yeah, nice study, you see. How are you going to quantify the PET?
Sorry, my connection is a bit unstable. Could you please repeat?
How are you going to quantify the PET images? Do you have arterial input function, or is it going to be reference tissue, or?
This is, David, very important question, and I'm sorry, but I'm not able to answer this one. I'm collaborating with Professor Laura Airas from Turku PET Center, and she has developed a study plan for the PET studies. But
okay.
Yeah, thanks.
So, and I had a question, and I thought Ragini may have a similar question as well. I wonder if you'd elaborate a little bit on the MRI procedures that you were planning on using, because obviously, I think that's going to be critically important to help identify some biomarkers.
Yeah, so we will of course obtain the conventional scans and also DTI, and we will also done sequences for NODI analysis, and I have planned the MRI scheme with our physicist, so it will be compatible with our previous studies. Of course, we will not be using the same scanners that we did 11 and 14 years ago, but they should be up to some extent compatible.
I was curious whether you were planning on getting like resting state functional connectivity data.
Actually, we have been discussing that, and at the moment they are included in the plan, but we don't have those done from the earlier studies, but we are planning to do them now.
Yeah, I would support that, and I'd be happy to talk to you further about some things we're doing.
Yeah, that would be very helpful.
It's very interesting.
Yeah, thank you.
Just a follow up. I'm sorry, I don't know how to raise my hand. This is Ragini on a cell phone. Hi, Jussi. Thanks a lot. Great work. I was just wondering, do you have a hypothesis on what imaging measures you want to actually associate with your metabolic panel?
My main interest will be DTI metrics.
I mean, some of the so the free water and all that will measure inflammation, I presume. But other than that, I mean, DTI has 1,000 metrics, right? So I would assume that. So have you done any study in the past which has looked at like a preliminary study in which you found some of them to be more associative than others?
With inflammation, no. We haven't actually, even though we have a PET center in our hospital, we haven't used it much. From our earlier study, we have PET images on a couple of patients, but they remain unanalyzed. Okay. So we don't have any preliminary data on that.
Thank you.
Thank you.
Well, again, thanks so much for sharing this study. And this was just launched, I guess, last year, right?
Yeah, last year we got the ethical permission, and we have now identified the living or the surviving patients, and we will now start contacting them. Our ethical committee did not let us to call them over the phone, but we need to send invitation letters. Let's see how that succeeds. Yeah.
You know, this study. So within the track TBI cohort, we're just about finishing with approximately a 5 to 7 year follow-up. Obviously, not quite as long as TBI care, and we have north of 300 TBI patients, together with another 100 or so sundry controls, that have come in person for a follow-up MRI, follow-up blood draw, etc. Obviously, no PET, no stool or any gastroenterological sampling, but we are interested in continuing to follow up on those people. And it'd be really interesting to, as we start analyzing some of the data, to connect with your team, right? Because this will be a very unique group of people, right? Where we have more than 5 year, and in some cases, I mean, in some TBI care project, you may have, 10 to 15 year follow-up.
Yeah, indeed. Yeah.
Yeah.
This is a great suggestion. Yeah, definitely we should join our forces. Yeah, definitely.
Okay. Peter, you have your hand up.
Hi, Jussi. Just curious, from the TBI care that you're doing the longitudinal follow-up, how many of the outreach are still alive?
It's a good question. So out from 200 patients, my research nurse Nina checked, and it seems that there are 122 patients alive who have imaging and biochemical data available.
And that's the biomarkers for admission day 1, 3, 5, 7.
Yes, and the follow-up visit at around 9 months.
Oh, okay, cool. Thanks.
And most of these people live in the kind of sort of Western Finland, Turku area.
Yeah, they live in our district still.
Your district, so that's terrific. They could be coming in. I mean, I think one advantage that we have within track is these 300 some odd people that we've identified live all over the country, right? They were
exactly
in one of 18 different sites. So that's a terrific opportunity there.
Yeah, indeed.
Okay. Does anyone else have any questions for this exciting project?
Just one question, Jussi. Did you manage to get funding, or are you still looking for funding for the study?
Yeah, actually, I managed to get funding, but the trouble is that I needed to return a quite big share from the acquired funding, because I received university position, and the foundation that funded me told me that the funding was meant for postdocs, not for professors.
Oh.
So I needed to return a big share, but they promised, or how would I say, they invited me to send a new application in the professor category next autumn, so I will do it.
We'll be collecting stool and blood and MR, but no PET in a cohort of patients with chronic TBI. We should probably chat offline as part of TBI Reporter. So we should see whether we can try and find ways to combine our data and try and collect common data elements appropriately.
Yeah, that would be really important too. And if it's possible for us as non-UK group to collaborate with TBI Reporter, I'm definitely interested in that.
Okay.
Absolutely. Any other questions from anyone on the conference? All right. Well, listen, thank you guys so much for logging in. And this is exactly the kind of information and the kind of knowledge transfers we need to have. I can sense that there'll be several important collaborations, very potentially very fruitful collaborations that will rise out of this. Okay. I do not believe we have anyone on the agenda yet for next month's teleconference. If anyone wants to, um, share some of your ideas and your data, please email me. If not, I will reach out and draft somebody. All right. Well, thanks so much, everybody. Really appreciate you logging on.